Hi, AP Bio folks. I just want to give you a little overview of the lab that we're going to be doing today. Today we're going to be collecting data for the Transpiration Lab, which is looking at the question, how do different environmental conditions affect the rate of transpiration in plants? If you were here last week, if you're a house B student, this should look familiar because you helped to set this up. But especially for those of you that are full distance, I wanted to give you an overview of how this lab is set up so that when you read it, it makes sense. What we did here is we took some plants, these are all about the same age, they grew in the same conditions, and we put them under various different environmental conditions. You can see they're wrapped in plastic bags so that the only way water can escape from here is through transpiration out the leaves. So any mass change that's happened in the last 5 days happened because water evaporated from the leaves. This picture shows the control set of plants. They're just sitting in the greenhouse in natural light. And then we took several other sets of plants. These are inside of this plastic container with a humidifier, so they have extra high humidity. Some of them were under this really intense light, which also added some heat. Some of the plants were sitting in front of a fan, so they were sitting in the wind for 5 days. And some were actually inside a cabinet, which was closed, so they were in the dark. And all of this was set up last Thursday, so they've been sitting there for about 5 days. Today is going to be a little bit of a unique day for you people at home because the main focus of today is to work on this lab, but the data won't be ready for you until this afternoon when 4th and 5th hour students collect that data. During the day right now, you should do the pre-lab part of the lab document that's attached to the classroom post today. It basically just asks you to analyze the experiment design a little bit and write your prediction for what's going to happen. Then you can work on whatever else you need to, finish your mastering biology homework that's due tonight or review for tomorrow's mini test. And at 3:30 today, once I get all the data from 4th and 5th hour, I will be attaching a spreadsheet to today's classroom post so you can access that data and finish up your lab. This lab isn't going to be due for a week so that you have time to ask questions, but remember, you can't ask questions until you start working on it, so don't put it off. Get started. Let me know if you have any questions, and have a great day.